Hello everyone, welcome to the biology section of our MCAT ACE lecture series. In today's lecture, we are going to describe amino acids, discuss their configuration and classification, describe how proteins are made of amino acids and how they can exist in different structure, primary, secondary, tertiary, and quaternary structure, and then finally describe enzymes, different forms of their functions, different conditions that can affect the enzymatic activity, and then finally go over the enzyme kinetics as well as inhibitors of different enzymes. And as always, we will conclude this lecture with some practice question. Now, with that outline in mind, let's start with describing the configuration of amino acids. So basically, proteins are polymers of amino acid. And what polymer mean is that a molecule that is made of a large number of similar units. So in this example, we have a protein which is a polymer because it's made of many amino acids that are bound together. Okay, so these are amino acids, and each amino acid here we can call them a monomer. So a polymer is made of many monomers that are brought together. Now there are 20 different amino acids that are out there, but all of them have a similar generic structure, which is shown here in this image right here. So all amino acids have an alpha carbon that is bound to an amino group on one end. So here we have the NH2, and bound to a carboxylic acid group on the other end. And since there is both an amino group and a carboxylic acid group, we call these structures amino acids. All right, and then there would be an H group also attached to a alpha carbon, and then finally we have a side chain that will be different for different amino acids. So all these 20 amino acids have the same amino group, carboxylic acid and hydrogen group, but it's their side chain that would be different. And here I'm going to show you different amino acids. So here you can see that all these amino acids that's shown here have the same generic structure. There's an alpha carbon, there's an amino group, there is a carboxylic acid group, there is an H. It's only the R group, which I've shown in orange here, is different for every one of these amino acids. Now, another point that I would like to point out here is that all these carbons, with the exception of the glycine, are bound to 4 different groups. So here you can see this alpha carbon is bound to 4 different groups. So for the rest of them. And so therefore, alpha carbon of all these amino acids, with the exception of glycine, are chiral carbon, which are optically active. And the reason glycine is not optically active is that its side group is an H. So since 2H is bound to this alpha carbon, it's not an optically active molecule. So back to the original image that we have here. Now later on in our lecture series, we discuss how DNA will be transcribed into mRNA, and then mRNA will be translated into protein. And so here mRNA from 5' to 3' direction is running through a ribosome. Its codons are being recognized by the anticodon of tRNA, and so every tRNA, depending on the anticodon that they're having, is bound to a specific amino acid. As these tRNAs are coming through the ribosome, the amino acids will be added to the growing peptide. Okay, now here I'm showing you how a peptide bond is being formed. We have one amino acid on the left side, another one on the right side. Once the NH2 combines with the COOH with the help of the peptidyl transferase enzyme, and H2O will be lost. So here we have a dehydration that is going on, as a consequence of which a C double bond O and H bond will be formed, which is an amide. And this peptide bond is kind of rigid because it has partial double bond structure as well as a planar structure, and it has a polarity. So here, if we have more amino acids added, you will notice that they all have the same direction. They are all from N to C direction, because here we will have an N to C bond. Here we have N to C bond. The next one would be N to C bond. So all these amino acids will have the N to C polarity or configuration. So here with mRNA having 5' to 3' direction read through the ribosomes, the polypeptide chain will be added from N to C terminus. The next concept is classification of different amino acids. So depending on their R groups, amino acids can have different characteristics and ionization properties, which can affect their structural as well as biological activities. So therefore, one very important approach to classify amino acids is based on the charge of their functional group. And so based on that, we can categorize amino acids into either non-polar or polar amino acids. So here we have the non-polar amino acids that have either alkyl or aromatic groups, and these non-polar amino acids are hydrophobic. So they are hydrophobic. Versus the polar amino acids, which are hydrophilic. And the polar ones itself can be divided into three groups. They're either polar uncharged, so here is an example of an uncharged amino acids, or it could be polar charged amino acids. Like for instance, in the example shown right here, we are having a positively charged amino acids that is a proton acceptor. So therefore, these are basic amino acids. And then next we have negatively charged amino acids like aspartic acid or glutamic acids, which are hydrogen donors. So these are now acidic amino acids. And I would like you to remember that aspartic acid and glutamic acid, these two are acidic amino acids and are negatively charged, versus lysine and arginine and histidine, these are positively charged and are basic amino acids. So if you need to take a minute to memorize these amino acids, please do so, because we will encounter them later on during this lecture. The next concept that I would like to discuss with you is called a zwitterion. So here we have a zwitterion. And Zwitter is basically a German word standing for hermaphrodite, where an individual has both male and female characteristics. So in chemistry, zwitterion refers to a molecule that has both positive and negative charge, but then the net charge of this molecule is 0. So with amino acids, there could be an internal transfer of hydrogen from carboxylic acid group to the amino group, as well as a consequence of which there would be a COO negative and an NH3 plus groups in this amino acid. So this is an example of a zwitterion, where there is both negative charge and positive charge within the same molecule, but the net charge is equal to 0. Now each of these groups, the amino group, has its own pK, which is about 9.0. So once we go above pH of 9, this amino group will lose its hydrogen. Same with the carboxylic acid group. It has its own pKa, which is approximately around 2. So once we go above pH of 2, the carboxylic acid group will lose its H+. And then the R group may or may not have a pKa. If we are talking about negatively or positively charged molecules, then those molecules also, those amino acids also have their own pKa. Now with that background information in mind, let's review the titration curve of different amino acids. So here, first I'm showing you a non-charged amino acids. And on one axis we have the pH, so from 0 to 12. And then on the other axis, we have equivalence of the hydroxyl group added. So as we add more hydroxyl group, the pH will increase, so the solution becomes more basic. So starting with the most protonated form of the amino acid here, you can see that we have an NH3 plus and a COOH. So it's a positively charged or a cation amino acids that we are having. As we increase the pH and we go beyond the pK1 of the COOH, therefore, COOH will lose its H+ and form a COO negative. So at this point now we have a zwitterion, because we have both a negatively charged and a positively charged groups in this molecule, but the net charge is 0. Now as we keep increasing pH, we will go beyond the pK of the amino group, as a consequence of which amino group will eventually lose its H+, and so it will turn into NH2. And so since there is only a negative charge here, this is an anion. All right. Now there is a point where at pK1 there is a 50% balance between the cation and zwitterion, versus at pK2 there is a 50% balance between the zwitterion and the anion. The point at which the amino acid is predominantly in the zwitterion form is the average of the pK1 and pK2. pK1 plus pK2 divided by 2. And this point is called isoelectric point. In other words, the particular pH at which a given amino acid predominantly exists as a zwitterion is called the isoelectric, or Pi. The next titration curve that I would like to discuss is for a basic amino acid. So you can see that in its most protonated form, the NH3 plus of the side group is also having a positive charge. And then we have the amino group with a positive charge and a COOH. So this is a 2+ molecule. Once pH goes beyond the pK of the carboxylic acid group, the carboxylic acid will lose its H, and now we have a 1+ charge. As pH continues to increase and we go beyond the pK2, now the amino side chain will lose its H+. So here we have a deuterion, or a neutral molecule, and then once we continue to increase the pH, again, the side chain will lose its H+, and we have an anion group. So here I'm showing you the pH curve. As we continue to add hydroxylic group, we will continue to lose H+ until we reach the deuterion form. So for this case, the isoelectric point is the average between pK2 and pK3, because think of it, at the pK2 level, there's a 50% balance between these two amino acid forms, versus at pK3, there's a 50% balance, there's 50% anion, 50% deuterion. So the pH at which the deuterion form is the predominant form of this amino acid is the average of these two. So pK2 plus pK3 divided by 2 will give us the isoelectric point of this molecule. And as we just discussed, this was a basic molecule, or a positively charged amino acid, and these were the list of positively charged. So the example that I've provided you here was actually lysine, with a chain of 4 carbon and 1 NH3+. The next molecule that I would like to discuss with you is a negatively charged amino acid, or an acidic one. All right. So here in this case, in the most protonated form, we are having COOH, and an NH3+. As we continue to increase pH, the COOH side group will lose its H+, and so here now we are having a deuterion, because we have one negative, one positive with a neutral charge. Now continue to increase pH, then the other COOH group will lose its H+, and then finally beyond the amino group pK, the NH3 also loses its hydrogen. So here is the titration curve for an acidic amino acids. We see that at pK1 and pK2, the two COOH will lose their H, as a consequence of which the isoelectric point is the average between pK of the carboxylic acid group and pK of the side chain. So pK1 plus pK2 divided by 2 will give us the isoelectric point, which is the pH at which the amino acid is predominantly in its deuterion form. Continue to add more hydroxyl groups to the solution, and finally we will have an anion beyond the pK3. All right, and so here is the list of negatively charged or acidic amino acids that we are having here, and the example that I gave you here was an aspartic acid. So with that background in mind, this is how we calculate the isoelectric point. I just described it for you, but let's go over it one more time. If the side chain does not have an ionizing group, like for instance in a glycine or leucine amino acids, then the isoelectric point is the average of the amino group and a carboxylic group. So the amino group we just described is pKa is about 9, the carboxylic group is pKa is about 2, so 9 plus 2 divided by 2, it's about 5.5 is the isoelectric point, generally for groups without an ionizing group. On the other hand, if you're talking about an acidic amino acid, like for instance aspartate or glutamate, then the Pi is the average of the side group and the carboxylic acid. So here we have the carboxyl group and the side group. The average of these two pKs will give us the isoelectric point for acidic amino acids. Versus if we have basic amino acids, it's the average of pK of the amino group and the side chain. So if we go back again to the second example, the pK of the side chain and the amino group is what gives us the isoelectric point. So now what I would like you to do is try to calculate the isoelectric point for these examples. All right. So what you can do is that you can pause the video. When you feel you're ready, you can proceed and watch the rest of this lecture. All right. So hopefully you have time to calculate the isoelectric point for these examples. So here the arginine, it's a basic amino acids, and so therefore 9 plus 12.5 divided by 2 would give us 10.75. So that would be the isoelectric point for arginine. Versus for aspartic acid, 3.9 plus 2.1 divided by 2 will give us number 3. All right, and so that would be the isoelectric point of the aspartic acid. The next concept that I would like to discuss with you is protein purification. So based on their isoelectric point, we can run proteins on a pH graded gel. So here you can see that we have a gel column where on top we have a pH of 12. As we go deeper, there will be a pH of 2. And then we have an electric charge that runs through this column. Here we have the cathode with a negative and then anode with a positive charge. Once we run proteins through here, what happens is that proteins based on their charge, whether they are positively charged or negatively charged, they will start to move on this column. But once they reach their isoelectric point, at that point they have a neutral charge, as a consequence of which there won't be any more movement. So here in this example, for instance, we have a basic amino acid or protein structure versus in lane C, we have an acidic one. And then finally in lane B, where isoelectric point is close to 6, we have a neutral amino acid or protein structure. The next concept is polyacrylamide gel electrophoresis. And so basically this polyacrylamide gel has matrix of small pores. And then once we run proteins through there, from one side running a negative charged cathode and then on another side a positively charged anodes, molecules that are negatively charged, those proteins that are negatively charged, has stronger affinity to move down the gel, versus the positively charged ones are having stronger affinity to stay up close to the cathode pole. Now at the same time, the larger molecules, because they have to move through the pores of the polyacrylamide gels, are moving very slow, versus the small molecules can easily, those small proteins can easily move through the pores. And so that's why here we can have isolation of proteins based on charge and size. So the small ones, which can easily move through the pores and are negatively charged because they are now more absorbed towards the anode, will move fastest down the lane. So here is where we've loaded our protein, and as it's running through this electric gradient, we can have proteins isolated based on their size and charge. So here we will have small and negative ones, versus the larger one have difficulty to move through, and those that are positively charged are also more attracted towards cathode. So here we will have the large and positively charged. And then the ones that are in between, that are either large or negatively charged, or are small and positively charged, would be in between. And then the final method of isolating proteins is based on using sodium dodecyl sulfate polyacrylamide gel electrophoresis. And basically this SDS is a detergent that breaks the covalent bonds. So it will break all the covalent bonds of a folded protein, and once it binds to the amino acid side chains, it gives a net negative charge. So all these positive and negative charges are now offset, and the protein, all it has is a negative charge. So the properties, the charge properties of a protein is now gone. And so if we run a protein on an SDS polyacrylamide gel electrophoresis, its isolation is only based on the size because the charge has been offset. So therefore, bands that are running farther down are smaller size proteins, versus the ones that are on the top are larger ones, which have difficulty to move through the pores, and so therefore they're moving slower. The next concept that I would like to discuss with you is the solvation layer or entropy. So basically solvation layer, this is referred to as structured organization of a solvent, like for instance water, that surrounds a solute molecule, like for instance a protein. So once we have a protein structure that is surrounded by many water molecules, if this protein have hydrophobic properties on its outside structure, then the surrounding molecules will have to form an organized structure to optimize the hydrogen bonding among molecules. And so therefore, since this is an organized structure, there is a low entropy. All right, so if the outside structure are hydrophobic, then there would be a low entropy as a consequence of which this protein structure is not very stable and is not favorable. Versus if we have a protein molecule that is having hydrophilic structure, hydrophilic residues on its outside exposed to the water, while the hydrophobic residues are inside the protein, there would be less disruption of water's hydrogen bonding, as a consequence of which now there would be high entropy. So if there are hydrophilic residues outside that is exposed to the water, there would be a higher entropy, and so therefore this protein conformation has more stability compared to the first example that we've given you. All right. The next concept that we would like to discuss is protein structure. So we've just discussed how the proteins are polymers of amino acids. They're made of many amino acids that are brought together. So this is a sequence of amino acids, which we can show it from N to C terminus. Now these molecules can form an interchain of hydrogen bonding between amino acids and form either a helical structure or a spiral structure we call alpha helix. So here you can see that there is hydrogen bonding between different groups. Or once two chains of amino acids come parallel to each other, they can again form hydrogen bonding between themselves, and this is called a beta-pleated sheet. So here you can see that we have hydrogen bonding that is formed. And then the beta-pleated sheet structure, if we have N to C and N to C structure, this is called a parallel beta-pleated sheet. Versus if we are having a protein like this, that we have N to C, and then here would be N to C, this is now an anti-parallel beta-pleated sheet structure. The next structure is a tertiary structure, and basically tertiary structure is a three-dimensional shape that proteins assume, and this folding and bending is being carried out by four major bonds, which I've just shown you right here. Number one, we have the ionic bonding. So number one, we have ionic bonding, which results from electrostatic attraction between positively charged residues, like for instance lysine, and negatively charged amino acids, like for instance aspartic acid. The next bonding is hydrogen bonding, which is formed between the highly electronegative oxygen and nitrogen atoms on one amino acids and the hydrogen atom that is attached to the oxygen or nitrogen of another amino acids. And so that would be the hydrogen bonding. Next, we have disulfide linkage, which can be formed between two cysteine amino acids that are brought together due to the protein folding. And this disulfide bond is very strong and stabilizes the tertiary structure. So here, let me show you a cysteine molecule. So here, we are having a cysteine molecule, and so a disulfide bond can be formed once two cysteine molecules are being brought together to form a disulfide bond. And then the final force is the dispersion forces, which arise when a nonpolar atom becomes momentarily polar, as a consequence of which there would be uneven distribution of electrons, and this uneven distribution will form a dipole that induces also a shift in the electrons of neighboring nonpolar atoms. Now these dispersion forces are fairly weak, but they could play actually an important role in the tertiary structure if the other forces, like for instance hydrogen or disulfide bond, is not present. All right, and then the final structure is the quaternary structure, which refers to proteins that contain multiple polypeptides that are brought together. So in this example, you can see that a green polypeptide is brought up with a red one. So this is an example of a quaternary structure. More than one amino acid chains are being brought together. And another example that I can give you is, for instance, hemoglobin, which is made of 4 chains of amino acids that are being brought together. Or another example that I can give you is gap junction, which is made of connexins. So multiple polypeptide chains are being brought together in order to form a quaternary structure. The next concept is discussing the conformational stability and folding. So we just described the forces that could help maintain a tertiary structure. But then there would also be forces that will disrupt this tertiary structure and cause denaturation of proteins. So here we would like to talk about factors that lead to denaturation of proteins. And these include, for instance, number one, heat or UV radiation, which will cause vibration of protein molecules, as a consequence of which there would be breakage of weak bonding, like for instance hydrogen and dispersion forces. And one example that I can give you is, for instance, in our body, most enzymes function best at around 37 degrees centigrade. If we have a fever and our body temperature goes beyond 42 degrees C, that's the time that there could be denaturation of proteins, and that's why fever of above 42 degrees centigrade can become lethal, because that will affect the function of proteins and lead to their denaturation. Number two, we can have organic compounds, like for instance ethyl alcohol, which is involved in intermolecular hydrogen bonding. So there will be hydrogen bonding, intermolecular form, that will disrupt the intramolecular hydrogen bonding. Next, we have salt of heavy metals, like for instance mercury and lead. So salt of heavy metals, which can disrupt the ionic bonds as well as disulfide linkages. So it can disrupt ionic bonds and disulfide linkages. Number four, we can have alkaloid reagents, which can disrupt ionic bonds by combining with positively charged amino acids in proteins. And then finally, pH changes can affect the conformation of a protein and lead to its denaturation by disrupting the ionic bonds. And one example that I can give you is that once we are eating a food that has protein, in our stomach there is a low pH, and so there would be denaturation of proteins. And once protein loses its three-dimensional conformational structure, that's the time that pepsin can now access peptide bond and cause denaturation of protein into smaller amino acids. Now, moving on to the next concept, which is the enzyme functions and classification. So basically, most enzymes are made of proteins. And the reason I say most enzyme is that there are some molecules, like for instance, ribozyme, which is an RNA molecule that has enzymatic activity. So not necessarily all enzymes are proteins. However, most enzymes are made of proteins. And so enzymes are basically having catalyst activity by decreasing the activation energy of a reaction. So here I can show you that we have the reactants that will have to be converted into products. And in order to do that, we will need an activation energy. Now, enzymes act as a catalyst and decrease this activation energy, as a consequence of which the rate of a reaction increases. So now back to here. So enzymes act as catalysts by decreasing the activation energy of a reaction. Now these enzymes are highly specific and can only bind to certain molecules. And those molecules that they bind to are called substrate. Now, in general, depending on the type of reaction that enzymes carry out, we can have 6 main types of enzymes. So the 6 enzymes that we have, number 1 is oxidoreductase enzyme groups, which are required to catalyze the oxidation reduction reactions. Then we have transferases, which catalyze transfer of a group to a molecule, like for instance kinase, which catalyzes transfer of a phosphate group. Then we have hydrolases, which mediate hydrolysis. And for instance, pepsin is an enzyme in our stomach that leads to hydrolysis of protein and breakage of them into smaller amino acids. Number 4, we have lyases, which will remove functional groups. So you know how transferases were adding functional groups, so here lyases will remove functional group. And one example is, for instance, the decarboxylase enzyme, which will remove the carboxyl group. Number 5, we can have isomerases, which will catalyze conversion of a compound into its isomer, like for instance an aldose converted into ketose. So isomerases will convert a compound, as the name implies, into its isomer. And then lastly, we can have ligases. So number 6, we can have ligases, which will catalyze bonding of atoms. Okay, and one example that I can give you is that if you have a DNA molecule and there is a nick in there, it's the job of a ligase enzyme to go in and join the 2 nucleotides together. Next, I would like to discuss cofactors, coenzymes, and vitamins. So we discussed how most enzymes are made of proteins. Now, some of these enzymes could be made of a pure amino acids and proteins, but some of these enzymes could have other helper molecules, and these helper molecules are called cofactors. So usually cofactors are inorganic ions, like for instance copper or zinc, that will help enzyme with its function. Now, if these helper molecules are made of only organic molecules, then we call them coenzymes. So these are helper molecules that are made of organic molecules. And an example of these organic molecules, for instance, is vitamins, which cannot be synthesized at adequate levels by our body, and so therefore we must obtain them through diet. All right, so these coenzymes can come in different forms, and these include, for instance, number 1, there are water soluble vitamins, like for instance vitamin C, which has antioxidant properties, and we can have vitamin B complexes, which are required for carrying out many different reactions. Now, the reason that these molecules are hydrophilic and are water soluble is that they have a large number of electronegative oxygen and nitrogen atoms, which engage in hydrogen bonding with the water, as a consequence of which now they're water soluble. The other form of vitamins that we are having are fat soluble. So number 2 is fat soluble vitamins, and these include, for instance, vitamin D, which is required for absorption of calcium and phosphorus. We can have vitamin E, which is similar to vitamin C, is an antioxidant. We can have vitamin A, which are required for our visual function, as well as for differentiation of epithelial cells, so skin and vision. And then lastly, we can have vitamin K, which is required for our coagulation reactions. Now, the reason these vitamins are fat soluble is that they have high proportion of hydrocarbon, and so therefore they are non-polar. Now, if you have noticed, here I've discussed how vitamin E and vitamin C are antioxidants. So what antioxidants do is that they will neutralize free radicals. Free radicals are basically uncharged molecules that are highly reactive and short-lived, because they have an unpaired valence electron, and so therefore antioxidants, by sparing an electron, will stabilize these free radicals. Otherwise, these free radicals, because they are highly reactive, they will react either with DNA or protein or lipid and cause damage. So these antioxidants are essential to prevent DNA or protein or lipid damage, which could lead to cellular aging. Now, oxygen-free radicals is a very important topic that could show up as a passage on your MCAT. So here I would like to discuss this in a little bit more detail. So when we are having an oxygen, and if we add H plus 4 electron, that's when two water molecules will be formed. And this reaction takes place physiologically in our electron transport chain for oxidative phosphorylation. We've discussed this later on in our lecture series. But what happens when we don't get 4 electrons in an oxygen? Oxygen-free radical will be formed. And so here we have an oxygen molecule. If we add only 1 electron to it, it will form a superoxide anion. And if we add 1 more electron to it, it will form a hydrogen peroxide. If we add 1 more electron to it, it will form a hydroxyl radical. And then if we add 1 more electron to it, then that's the time that water will be formed. So now, can you tell me, of all these molecules, which one is the most dangerous and most toxic one? And the answer is hydroxyl radical, because this is the most reactive one. This is also the most damaging of all these. So one mechanism of forming all these reactive oxygen species is physiologically through our electron transport chain, because as NADH and FADH2 transfer electron to the oxygen, these reactive oxygen species will be formed. But then there could also be pathologic formation of these oxygen-free radicals. Like for instance, once there is inflammation in our body, some of our defense cells will intentionally create oxygen-free radicals in order to kill pathogens. So one mechanism is by inflammation. There is also ionizing radiation that can lead to formation of oxygen radicals. We can have heavy metals, like for instance copper and iron, that acts as Fenton catalyst and lead to formation of free radical. And so therefore disorders like for instance Wilson disease that have excess copper formation, or hemochromatosis for excess accumulation of iron, these patients are at increased risk of oxygen-free radical damage. And then lastly, there are drugs and chemicals that will lead to the formation of oxygen-free radicals. Now there are multiple defense mechanisms against these oxygen-free radicals, and we have already discussed one of them, which was the antioxidants. So let's go over them one more time. So now let's discuss the defense mechanisms. Number one, we can have antioxidants, like for instance vitamin C and vitamin E, that will defend against these oxygen-free radicals. Alternatively, we can have metal binding proteins, like for instance transferrin, which binds to iron, or ceruloplasmin that binds to copper. And so therefore, by taking away these heavy metals that could be involved in Fenton catalyst reaction, we will decrease the risk of free radical formation. So metal binding proteins. And then lastly, we have enzymes that have antioxidant properties. Like for instance, one very important enzyme is the superoxide dismutase. So superoxide dismutase is involved in conversion of superoxide anion into hydrogen peroxide. Now I would like to point out that this enzyme is not purely made of protein. It actually does have some metal cofactors. So you know how earlier we discussed that some of these enzymes are having cofactors. So superoxide dismutase is one example that relies on the help of some metal cofactors in order to convert superoxide anion into hydrogen peroxide. And then the other two important enzymes are catalase and glutathione peroxidase, which are involved in conversion of hydrogen peroxide into a hydroxyl radical, which is now one step closer to be neutralized and be converted into water. All right, next moving on to the enzyme mechanism of action. So once we are having an enzyme, we discussed how this enzyme is pretty specific in binding to its substrate. So we can have an enzyme added to a substrate. This reaction is bidirectional, and it can form an enzyme substrate complex. Now, this enzyme substrate complex can now yield the enzyme plus the product. So the cleft or the pocket at which the substrate binds to the enzyme is called the active site. So here is the active site of this enzyme. And initially it was proposed that the active site has the exact same conformation as the substrate. In other words, it's similar to a key that fits into a tumbler lock. And so that's why the name lock and key model was given to it. But then later on, it was shown that the enzyme actually changes conformation. Its active site changes conformation so that it fits the specific substrate that it binds to. So there will be change in the conformation. Here you can see how the enzyme has closed on itself in order to fit its substrate. And so therefore, later on, a new model called induced fit model has been proposed, stating that the active site forms a shape complementary to its substrate. The next concept is the allosteric enzyme. So basically, allosteric enzymes are enzymes that change their conformation upon binding of an effector molecule. And the allosteric site is a site different than the active site. So in this very first example, for instance, you can see how once an activator molecule binds to the allosteric site, the active site will now change conformation so that it can bind to the substrate. Or alternatively, instead of allosteric activation, there could be allosteric inhibition. In this example, the enzyme can bind to the substrate as is, but once the inhibitor binds to the allosteric site, the active site will now change conformation to a form that the substrate can no longer binding. So the allosteric site can cause either allosteric inhibition or allosteric activation. And this concept is very important for feedback inhibition. So you know how in biological reactions, we have one product being converted to another product with the help of, let's say, enzyme X. But we don't want too much product B to be formed because A is an important, valuable substrate and we don't want to use it all up to the product. So what happens when we have accumulation of the products? Sometimes for certain enzymes, these products can act as allosteric inhibitors and go and bind to the enzyme that has formed it and inhibit it. So one mechanism of not letting too much products being formed is by feedback inhibition, where product of an enzyme will act as an allosteric inhibitor for that enzyme. The next concept that I would like to discuss is conditions that will affect the rate of an enzymatic activity. So you know how enzymes, most of them are made of proteins, and so this is the three-dimensional structure of a protein that contributes to their enzymatic activity. So it's important that the three-dimensional structure is maintained. If you disrupt that structure, therefore the enzymatic activity will also be inhibited. And so this is the concept that we are discussing here, how temperature and pH and other factors can affect the rate of an enzymatic activity. So effects of local conditions on enzyme activity. Number one, substrate concentration is one factor that affects the rate of a reaction. Because imagine we have like thousands of enzymes, but we only have one substrate. This enzyme is very limited because we don't have any substrate here. But as we add more and more substrate, these substrates will bind the enzymes and the rate of the reaction will continue to increase until all the enzymes are saturated, and then at that point we have reached our maximum enzymatic reaction rate. So here the substrate and the reaction rate, as we add more substrate, the reaction rate will continue to increase until all the enzymes are saturated. So here we've reached a saturated point. And so this is a Vmax, maximum rate of a reaction, because all enzymes are saturated. Now, in this situation, the only way to increase the rate of a reaction is to increase the number of enzymes we are having. So if we have unlimited number of substrate, we can increase the rate of a reaction by adding more enzymes. So in this case, the rate of a reaction will increase like this as we add more enzymes. All right. And then finally, we have temperature and pH that can affect the conformational structure of an enzyme, as a consequence of which they could disrupt the enzymatic activity. So in our body, for instance, it's at 37 degrees centigrade that most enzymes are having optimal activity. So here at around 37 degrees C, we are having best optimal enzymatic activity in our body. As we increase the temperature or decrease the temperature, the conformational changes will interfere with the enzymatic activity. So that's why once we go beyond above 42 degrees centigrade, that's when it can become lethal, because the enzymatic activity can be affected. Same with the pH. pH can affect the ionic bondings of the enzyme and the conformational structure. So that's why at a neutral pH is when most of our enzymes are functioning optimally. So here at a neutral pH of around 7 is where most of our enzymes are functioning the best. All right, next concept is the enzyme kinetics. We just discussed how enzymes increase the rate of a reaction by decreasing the activation energy. Now, as we add more substrate, so as we increase the substrate concentration, the rate of a product formation will also increase, as a consequence of which the velocity of the reaction will also increase until we reach a maximum velocity, Vmax. Now at this step, all the enzymes are saturated. So enzymes are saturated. And regardless of how much more substrate we add, the rate of a reaction will not increase. So in this case, the only way that we can increase the rate of a reaction is to add more enzyme. Now with this concept in mind, next I would like to describe the Michaelis-Menten equation for you. So this is a very important concept. It's called Michaelis-Menten equation, which is shown right here. And basically this equation can predict the rate of the enzymatic reaction. So here we can predict the rate of a reaction as a function of its Vmax and the substrate concentration. So if you look at this curve, we have reaction velocity on one axis, substrate concentration on the other axis. As we add more substrate, Vmax will be reached, because here at this point, all the enzymes are saturated. Now the concentration, the substrate concentration at which the V is at half, so here you can see the velocity at this point is half of the Vmax. This is called Km, or Michaelis constant. And in this equation, you can see that Michaelis constant is in denominator and can be added to substrate concentration. So the unit for Michaelis constant is the same as substrate concentration. So that's the first important concept. Unit for Michaelis constant is substrate concentration. And in fact, if you plug in this value in this equation, what you get is that V0 is equal Vmax times substrate concentration divided by substrate concentration plus substrate concentration, which is equal to 2S. So 2S. And once you cancel out the S together, V0 is equal to Vmax divided by 2. So here, what I've just shown you is that Km is the substrate concentration at which the speed of the reaction is half of the Vmax. So here Km is the concentration at which our speed of the reaction is at half of Vmax. So that's the second important concept. At concentration Km, rate of reaction is half of Vmax. And then the last concept that I would like to discuss with you is the enzyme affinity for the substrate. So if the enzyme affinity for the substrate is high, these enzymes will bind substrate very fast and the Vmax will be reached at a faster speed. So here you can notice that the Km has been decreased for this enzyme. So number 3 concept, so number 3, as Km decreases, the affinity of enzyme for substrate increases. And the reverse concept also applies. As Km increases, there is a decrease in affinity of the enzyme for the substrate. And here let me show you with another example. So here we have another enzymatic reaction that it takes longer for the Vmax to be reached. So here the Vmax is reached at a higher concentration of substrate. So here the Km is higher. So for this example, since the Km is higher, it tells us that the affinity of the enzyme for substrate is lower. All right. So three important concepts came out of this Michaelis-Menten equation. Number one, the unit for Km is substrate concentration. Number two, at concentration of Km, velocity of the reaction is half of the Vmax. And number three, Km indicates the affinity of the enzyme for the substrate. When Km is low, there is high affinity, and when Km is high, there is low affinity of the enzyme for the substrate. Now here, what I would like to remind you is that we are talking about Michaelis-Menten equation on a hyperbolic curve. So this is a hyperbolic curve, which assumes that there is only a single binding site for the enzyme and there is no cooperativity. But then there are also enzymes that have multiple binding sites. So there is multiple binding sites, and then there is also cooperativity, which means that once a substrate binds to the enzyme, binding of the rest of the substrate to the other sites of the active site of that enzyme is enhanced. And so therefore these enzymes will have a sigmoidal shaped curve. So this is a sigmoidal shaped curve that we are having. And this sigmoidal shaped curve is not being applied to the Michaelis-Menten equation. Michaelis-Menten equation is only for these hyperbolic curves, which is assuming a single binding site with no cooperativity. The last concept that I would like to discuss with you is different modes of enzyme inhibition. So number one, we can have competitive inhibitors. And these competitive inhibitors, what they do is that their structure is very similar to the substrate. So you know how we discussed that enzymes are specific to the substrate that they're binding, but sometimes there are some molecules that have a similar structure and the enzyme can confuse them as their substrate. And so there could be competition between the substrate and those inhibitors. So these inhibitors resemble the substrate and bind to the active site. As a consequence of which, since they are binding to the active site, the substrate can no longer bind to the active site. So the affinity of the substrate for the enzyme decreases. And so, as we just discussed, when there is low affinity, there is an increased Km. But then if we continue to add more and more substrate, we can actually overcome and compete with the inhibitor and reach the Vmax. Because imagine, if we have 1,000 enzymes and we have like 10 or 20 inhibitors, if we add 1 billion or 1 zillion substrates, these inhibitors don't have a chance to compete with all these substrates. The substrates will bind to the enzymes, and so therefore Vmax could be reached. So here, I'm showing you an example of a competitive inhibitor. The substrate and the inhibitor both have a similar conformation, and so therefore they're both competing for the active site. And so what happens when we are adding a competitive inhibitor to a solution? Km of the reaction will increase, so there is increased Km. The new Km is at this point right here, but then the Vmax could still be reached as we are increasing the substrate concentration. All right, so with that information in mind, now we can fill in this table. So the competitive inhibitors, they're binding to the active site. They would increase Km, but Vmax is still the same. There is no change. If you add more and more substrate, you still can reach the Vmax. The next form of inhibition is non-competitive, and these are basically binding to the allosteric site, and so they are not competing over the active site. So once they bind, they change the conformation of the enzyme, and substrate can no longer bind to the active site. So regardless of how much more substrate you're adding, it cannot compete with the inhibitor, and so therefore Vmax would be reduced. But the affinity of the substrate and enzyme is still the same. All right, so Km has not been changed for this type of inhibition. So here I'm showing you a non-competitive inhibitor. Once the allosteric site is bound by the inhibitor, the substrate can no longer bind to the enzyme. The affinity is still the same, so Km will remain the same, but since the enzyme is non-functional, the rate or Vmax of the reaction has been reduced. So in this example, we will have something like this. All right, so the Vmax has dropped from here to there, but Km, where half of the Vmax is reached, is still at the same concentration of substrate. So for non-competitive inhibitors, they bind to the allosteric site. There won't be any change in the Km, but the Vmax would decrease. And then the final form of inhibition is called uncompetitive inhibitors, where they bind to the enzyme substrate complex. So you know how we discussed enzyme plus substrate will give us enzyme substrate complex, and this can be converted into enzyme plus product. This type of inhibitor is binding to the enzyme substrate complex. So once it binds to it, the enzyme can no longer release the substrate. So in other words, the affinity of the enzyme for the substrate has been increased, and so Km in this case would decrease. And then at the same time, since the product can no longer be formed, the Vmax would also decrease. So here in this example, the enzyme is bound to the substrate. Once the inhibitor binds to the enzyme substrate complex, the enzyme can no longer release substrate. So Km has now decreased. And then the product could not be formed, so Vmax is also decreased. And then if I want to show it to you on this curve here, it would be something like this. So Vmax is dropped from here to here, and then Km is somewhere around here. Okay, so that's our new Km. And so for uncompetitive inhibitors, they bind to the allosteric site, they decrease Km, and they decrease Vmax. All right, and that concludes our discussion of the amino acid lecture. Next, I would like to review some practice questions with you. So what I would do here is that I will read the questions, then give you a few seconds to pause the video so that you can answer the questions by yourself, and then when you're ready to go through the answers, we will solve the questions together. All right, so question number 1. A solution of 3 amino acids are loaded into a gel with a stable pH gradient and an electric field. Which of the following amino acids are localized in lane A, B, and C? All right, so we just discussed this earlier in our lecture. This is isolation of amino acids based on their isoelectric point. We have an electric field and we have a pH gradient. All right, so that's question number 1. If you want to pause it and then answer the question, then go ahead by all means. All right, hopefully you had enough time to review this question. So now let's review the answers here. So what we are having here is a neutral amino acid, an acidic, and a basic one. And here I've provided you with a graph. Glycine has an isoelectric point of 6, about 6, 5.97. Arginine has isoelectric point of 10.8, and aspartic acid has isoelectric point of 3. So by not having this curve, we would remember that we have positively charged or basic amino acids like lysine, arginine, and histidine. So arginine is a positively charged or basic amino acid. And then we have the negatively charged amino acids like glutamic acid or aspartate, glutamic acid and aspartate. So what happens with the basic amino acids is that they stop at lane A, while acidic amino acids will stop at lane C. And then the neutral amino acids like glycine will stop at lane B, because that's corresponding to their isoelectric point. And so here I've provided you with a reminder. So lysine, arginine, or histidine, these are basic ones. They would start at lane A. Aspartic acid and glutamic acid will stop at lane C, and then the neutral ones will stop at lane B. All right, so with that information in mind, now let's answer this question. Where does lane A, what does lane A contain? It has to be a basic amino acid, and these include, again, arginine, histidine, or lysine. Of all the answer choices, these two so far are correct. Then for lane B, which is a neutral pH, we can have glycine, so choice D is correct so far. And then for lane C, we will have acidic amino acids, so choice D would be the correct answer. All right, next question. Which of the following changes are likely to be observed upon addition of competitive inhibitors to an enzymatic reaction? All right, so this question should be now easy to answer after we reviewed this lecture. So go ahead, pause the video. When you feel you're ready, you can restart the video. All right, so we just discussed how competitive inhibitors are competing with the enzyme over the same active site. So here we can have an inhibitor which has a similar conformation to the substrate. All right, and so the rate of the reaction and the substrate concentration as we add, so this is the original reaction, as we add the inhibitor to it, the reaction will be changed something like this. All right, so Vmax still will be reached, but at a higher substrate concentration. So what happens in this case is that Km would increase, but Vmax remain the same. So choice A is incorrect because Vmax is not changed, so is choice B, and then the correct answer here would be increased Km. All right, so choice C would be the correct answer. All right, and that concludes our first lecture on the biology section of the MCAT ACE lecture series. I hope you found this lecture helpful, and we will see you for the next video.